Hello, my name is Olivia Bird, and I will be discussing the structural analysis of solid electrolyte lithium batteries. So, these sorts of batteries have often been considered a next generation of lithium battery. They use a pure lithium anode, and as the name implies, they also have a solid electrolyte, such as ceramic or polymer materials. For our study, we're using a ceramic electrolyte with high ionic conductivity called beta Li3 PS4, hereafter referred to as LPS. The advantages of this sort of battery is they have a greater voltage window because of that lithium anode, which would not be stable with a conventional liquid electrolyte, such as lithium-ion batteries, improved safety due to not being filled with flammable liquid electrolyte, and the potential for a longer charge lifetime. However, they are not currently technologically viable due to certain failure mechanisms. One failure mechanism that has been observed is delamination, which is when the electrolyte and the electrode lose contact and thus can no longer conduct. However, the one we'll be most focused on for this study is the growth of lithium dendrites. When you pass current through lithium metal, it has a phenomenon known as dendritic growth, which is where it grows small spikes outside of the lithium layer. If these spikes get into our electrolyte, it can cause decomposition and reduce the effectiveness of said electrolyte. If they push all the way through from the anode to the cathode, as seen here, this can cause a short circuit. So studying these is important. As these dendrites occur within a solid material, which we cannot readily disassemble without breaking them, we have to use X-ray tomography to appropriately study their morphology. So to determine where dendrites grow, we take a look at our X-ray scans. The dark region is something which is X-ray transparent, meaning it could either be empty space or lithium, as lithium is pretty X-ray transparent and our LPS is not. As we cycle our battery, we can see that the crack of lithium or X-ray transparent material grows significantly. If we want to quantify this growth, we used a machine learning process which identified lithium and LPS and made them all one color. With this quantification, we were able to determine that when we didn't cycle it, this region was 17% lithium/void by volume, and after 2 cycles, that grew to 26%. We can also see that there were initially 23 separate regions of lithium, a region being a connected area in 3 dimensions. So this big crack here would be one big region, this spot here would be a really small region. You can obviously see other regions throughout there. And the number of regions decreased to only 11 afterwards, even though the total amount of lithium increased. So we also want to look at what happens outside of that sort of crack that we chose to look at really specifically. So here's the region we were looking at before, and we chose it because of that really obvious crack growth. The rest of this region is just the arbitrary area around that lithium. So we want to see what happens in that outside area. We see the same trend of increasing lithium and decreasing regions, but both changes to a much smaller extent. So these 2 slides list the conclusion that lithium growth does occur throughout the entire cell, but is primarily focused to certain localized regions with existing defects. That's because the mechanism for lithium migration isn't creating new cracks, but is instead infiltrating the existing regions and causing them to expand and breaking apart the electrolyte through that way. So from this, we can conclude that lithium dendrites grow by migrating into existing voids in solid electrolyte. Lithium migration is not evenly distributed and tends to be concentrated in localized areas with existing defects. And in future research, we'd like to determine how conditions such as the current density, how we make the LPS, temperature, etc., will affect our lithium migration. We also learned that there's a lower limit of quantification. We tried to do a second ROI shown here, and because of the size of this, our Weka segmentation was not able to appropriately identify the difference between lithium and LPS, and we couldn't get good data. We'd also like to continue to automate our process of identification and quantification so we can improve efficiency and repeatability. I'd like to thank my mentors, Professor Svetlana Pylapenko and Natalie Seitzman, as well as the Mines Undergraduate Research Fellowship for my funding. I'd also like to thank you for watching, and I wish you a good day. Thank you.